Any questions before we leave? So we're just going to kind of get into hematology a little bit. And again, then we'll do our dissection, and that'll leave the 2 days to kind of go over the rest of hematology, kind of take a nice approach to the physiologic components to it. And again, I'm going to keep saying this, if you talk to somebody who is taking this from somebody else, there are instructors that will put this together with what you call. I'm going to start talking, so you might want to pull that printer open. Just pull the drawer open. So they will continue that process. So hematology and the immune system really don't play well, or there's too much information. So I'll be curious to see once we get done with the term if you really thought that adding hematology to the immune system is a benefit, because it really is not a benefit. Now anytime you see the word hemato, heme, hemo, these all have to do with blood. Okay? And we'll kind of abbreviate that as we go through, because we're going to be talking about the different types of blood cells that are present there. Now, blood, so if I was to take, and we used to actually do this in the lab until we had all of our bloodborne pathogens and so forth, and then we were required to stop. But if I was to take blood from somebody and put it in a capillary tube, and I spin it down, okay? When you do this particular process, you allow for the heavier things to go down towards the bottom, and you allow for the lighter things to be at the top. All right? So this is what you end up seeing. You have what's referred to as the formed elements, in other words, structures, and then we have the liquid part of blood, which is our plasma. Okay? Now, this is going to be a chapter where you're going to have to know roughly where numbers are, okay? Not specific. This is going to play a big role as we start to move into the other term, because when we get into the renal system, you're going to see that this plays, again, a fairly large role when we calculate the amount of blood that gets filtered, okay? So this is the liquid part, and you can see that it's about 55%. So of your blood, a little over half is considered the liquid part of blood. Now, when we talk about lab work, okay, like a chem screen, if you've ever heard anybody have a chem screen or a mini chem, this is where we look at things like our electrolytes, so our sodium, our potassium, our chloride, our calcium, our magnesium, our phosphate, those types of things. We're looking at the plasma component. When we look at the amount of oxygen that we have in our blood, I'm going to amend that a little bit as we move through this lecture. The amount of gas that's dissolved, okay, it comes from the plasma. The amount of carbon dioxide that's dissolved is going to come from the plasma. Now, everything else, our blood cells, both our white blood cells and our red blood cells and our platelets, are essentially going to come into this area here with our formed elements. Now, the formed elements are then divided into this little tiny part here called the Buffy coat. And this is essentially where we have our white blood cells, because when we look at the numbers, we're going to see they exist at a much, much lower rate than our red blood cells. And then we have the large amount of our red blood cells here. Okay? Now, there's going to be a couple things I'll go over, and I'll come back and talk about it. But since this picture's up here, one of the things that we derive from this is a thing called a hematocrit. The other thing that you're going to notice as you start to work in the healthcare field or surrounded is that we're very lazy people. So instead of saying hematocrit, it's not uncommon to say the patient's crit is this. So we get really, really lazy and just do that. Not a smart thing, because one of the things that you find in the healthcare field is things that, you don't have to write this part down, things that mean, are represented by the same letters that mean two totally different things. This is mean arterial pressure, this is mean airway pressure. Vastly different. Okay? So, again, probably always good to call it by its full name. So the hematocrit is essentially the amount of red blood cells per the total volume of blood itself. Okay? So again, when we do this in labs, we do capillary tubes, seal them on both ends, and then you spin them, and then you measure the amount of red blood cells, and then you take the red blood cells and you base it off of the total amount that's here. Okay? And we get a value depending on your gender, which is the most significant characteristic, then followed by things like your athletic ability, not necessarily ability, but your athletic routine, because people will have this change dramatically, to your diet. Okay? So women can have a hematocrit that's, 43, 42, somewhere in that general ballpark, up to about 46 to 47. Men can have one that's going to be higher, about 48 to about 52. Okay? And again, take-home message, you should have an idea what a hematocrit is. You should also have an idea about how blood is broken down. Now, blood itself. So even though we have these formed elements, and I also have my plasma, it has 3 primary functions. Okay? First one is transport. It's a mode of taking nutrients to the tissues. It's your highway. That's how we have distribution. Not only can I take nutrients, so if I'll use this analogy in hematology, I'll use it in cardiovascular physiology, and I'm also going to use it when we get into renal physiology and ANP3. But if this is a flight, okay, and I'm a flight attendant, and I'm bringing you nuts, pretzels, drinks, what am I going to have to do probably in about 30 minutes? Pick up the trash. So not only do I take stuff to the tissues, but with all metabolic processes, I'm going to have wastes. So I'm going to have to pick the waste up, the trash, and take it to the appropriate area of the body to dispose of it. Okay? I take my oxygen to tissues. So on the very first day when we talked of AMP1, when we talked about the importance of our systems and how they work together, so if I have 1 mole of glucose without oxygen, I'm going to get, and again, depending on your biochemistry textbook, 2 to 4 moles of ATP. If I use the same process, but I make it with oxygen, so again, people aren't familiar with the abbreviation, so to speak. An S with a line over it is without, a C with a line over it is with. Okay? So with oxygen, I'm going to get 32 to 36 moles of ATP. And again, there's a range in there because depending on your biochemistry textbook, the biochemist that you're talking to, we add things up a little differently. But what's the take-home message? With oxygen is a much better way of making ATP. That's the reason why we use our blood to transport oxygen. This is a waste. I have to get rid of that. So an equation that you're going to get very, very tired of seeing, a little bit in this term and a lot next term, and you do not have to write this down. So carbon dioxide plus water gives me this really unstable acid called carbonic acid, and then I end up with bicarbon hydrogen ions. Hydrogen ions is equal to the negative log of the hydrogen ion concentration, which is just a really fancy way of saying The more I have of this, the lower my pH. When I change my pH, I change functions in the body, not in a good way. Okay? So when I make ATP, I get those two things. Well, I can use water. If I don't get rid of carbon dioxide, I end up with hydrogen ions, and I end up with a lower pH. So carbon dioxide should always kind of stick in the back of your head is a waste. It's not a good thing for our body. It's the reason why when we exhale, carbon dioxide leaves. I'm not exhaling a huge amount of oxygen. I'm exhaling more carbon dioxide. Okay? Now, the other thing that's in there, hormones. If you go back all the way to NP1, we talked about two different types of glands, exocrine glands and endocrine glands. Exocrine glands manufacture something on the inside, so if I'm an exocrine gland right now, I'm going to manufacture something and I'm going to put it on the surface of my head. It's going to be some sort of lubricant, what have you. Okay? If I'm an endocrine cell, an endocrine gland, I'm going to manufacture something on the inside and I'm going to put it in a little package and I'm going to stick it in the mail and it's going to go far away and it's going to affect cells in another part of the body. It's the definition of a hormone. How do they travel? Blood. We have regulation. Okay? Body temperature. Blood is generally 1 degree warmer than the rest of the body. That allows for me to take heat to the skin, and it allows for heat to leave my skin so we cool off for a variety of different ways, whether it's convection, whether it's radiation, whether it's condensation, evaporation. Not super important, but just know that that's how we regulate our body temperature. I regulate my pH. pH, again, is our acid base. The normal, the range of pH in blood, we're going to be talking about arterial blood. Venous blood is slightly different, but arterial blood, our pH is 7.35 to 7.45. So whenever I have this increased amount of hydrogen ions, the blood has to have a way of sopping them up. So the example that I used with the photoreceptor, how when we use the photoreceptor, its interaction with the bipolar cell. The bipolar cell was the 4-year-old. What did the bipolar cell want to do? Wreak havoc. But as long as the bipolar cell was turned on, as long as mom and dad were there, nope, you're going to stay right next to me. But if mom or dad decides to reach over and start doing something over here and no longer playing around, in other words, I'm turning off the photoreceptor, the bipolar cell goes crazy. Okay? Hydrogen ions are the same way. It's free hydrogen ions. Nothing that's bound. So I have proteins in my blood that basically attach themselves to hydrogen ions. Therefore, it doesn't affect my pH. Why? Because they're bound to something else. It's only the little 4-year-old running around by himself that's the problem. But as long as I've got a hand on the 4-year-old, the hydrogen ion, we don't have an issue. Okay? They maintain blood volume. And what I mean by blood volume is I keep a certain amount of plasma with all my formed elements. Because when that goes down, my blood pressure goes down. My blood pressure goes down, then blood doesn't go round and round. And remember, to stay alive, air goes in and out, blood goes round and round. It's just that simple. So these proteins act as water magnets. They hold water in. Plasma, you're going to find out when I talk about it in just a second, is mostly water. So that's how it maintains our blood volume. And then we have protection. Okay, we're going to talk a lot about clotting. Clotting is called hemostasis, the stopping of blood. So when I have a tear in a blood vessel, I don't have my blood leaking out. Because if it leaks out, it's not going to be able to have blood pressure and I can end up dying. And then we'll spend the whole entire last part of the term talking about our immune system. Now, if you haven't really figured it out, this is probably an excellent test question. What are the functions of blood? And give me a couple of examples. This is how you should always be thinking when you're either listening to a lecture or when you're looking over your notes. If there's a list of something, that's a really good place to start with a possible test question. You know, how many cranial nerves are there? And can you name them? And what are their functions? So So what are the functions, the 3 functions of blood? And give me an example of each of them. That'd be an excellent test question. Any questions on this? So what are some of the characteristics of blood? Okay? So I already told you that blood is basically 1 degree warmer than the rest of the body. So our body is 37 degrees Celsius. Did you have a question?
I was going to ask if it's 1 degree Celsius or Fahrenheit, but you answered it.
Yeah, it's Celsius, and you can figure it out if you go through the equation what the difference is, but it's not too far off from the Fahrenheit standpoint. So it's and again, is it important to know that it's 1 degree? The importance is that blood tends to be warmer than the rest of the body, which allows for heat transfer out. Okay, that's how we regulate it. pH 7.35 to 7.45. Sometimes you're going to hear, what is the average? Well, what's halfway between 7.35 and 7.45? 7.4. So sometimes you'll see people say the normal pH of blood is 7.40. Okay? Understanding the range sometimes is a little bit easier, simply because you then know where you're outside of your range. The volume of blood. Okay? So you basically have, depending on your height, your gender, you basically have about 5 to 6 liters of blood. Okay? Roughly about 8% of your body weight in kilos. So obviously, if you compare me to Shaquille O'Neal, I probably have 5, 5.5 to 6. He probably has 6 to 7 to 8 liters of blood. Okay? Much larger individual. Blood tends to have a very sticky component once it comes out of the body and starts to dry. So if you've ever seen your own blood, or if you've been part of an unfortunate accident where you've been exposed to blood, it does get a very sticky quality to it, which is some of the components we're going to talk about when we get into blood clotting. Now, I told you that we have 2 components to it. We have the liquid part of blood, which is plasma, about 55%. And again, just to make it super easy for you, about half of our blood is plasma. So if we talk about 6 liters, then about 3 liters of my blood is plasma. And what is most of it made of? Water. Any liquid in our body, for the most part, is going to be predominantly water with stuff added in. Whether we're talking about our aqueous humor, our vitreous humor, our cerebrospinal fluid, breast milk, ejaculate, everything is predominantly water. Just different stuff added in. Okay? So here, what are we going to add in there? Proteins. Okay? This is where most of proteins exist, is in the plasma. Most of these proteins are actually manufactured in the liver. So always keep in mind that if you're taking care of a patient that has liver disease, there's a big chance that they might have a very low level of proteins in their blood. Well, what did the proteins do? They help with our acid base. That was that whole thing over here with regulation, and they also maintain our blood volume. If I don't have proteins in the blood, remember proteins were water magnets. If I don't have enough of them, then the water part of our blood, the plasma, tends to leak out. It's one of the reasons why you see people with liver disease that might have kind of a distended belly. It's not that they're fat, it's that they have more fluid in their peritoneal cavity. Okay? Because it's not staying in their blood vessel because they don't have enough protein. Albumin is probably the biggest protein that you're going to see. The next ones have to do predominantly with clotting and with our immune system, our globulins. Okay? We're going to see our electrolytes. And again, sodium, potassium, calcium. Things that when I put in solution, they dissociate and they carry a charge. That's why they carry the term electrolytes. Because they carry a charge, therefore they conduct an electrical impulse. So really, when you talk about the fact of, if I was to take pure water and I was to drop something electrical in there, so the whole bathtub radio thing, okay? If it's just water, sterile water or distilled water, it's not really going to conduct a charge. The reason it's so detrimental is because when you get into water, all of your surface, so we sweat, our sweat has a lot of salt on there, which is an electrolyte, and that starts to conduct a charge. okay? So water in and of itself, as long as it's distilled and has nothing else in there, really doesn't conduct a charge that well. And then we have our respiratory gases. The respiratory gases specifically I already talked about, it's going to be oxygen and carbon dioxide. We're going to be taking oxygen to the tissue so that we can have this process, and I'm going to be picking up carbon dioxide and taking it back to the lungs so that I can exhale it, so I can control the amount of hydrogen ions that are present. Any questions so far? Now, I'm not going to get too far into this component because it's probably a good place to start tomorrow, but I do want to talk about, just to introduce it to you, we have red blood cells, which are referred to as erythrocytes. You ever heard the term erythema, erythema? When people, if you're describing the color of their skin, it's a very red skin. Now, what I want you to notice is when you look at the numbers up there, anytime we talk about red blood cells, women will always have a lower number than men. Okay? For two primary reasons. The biggest one is testosterone. Testosterone actually increases the activity of another hormone, which I'll talk about tomorrow, that causes red blood cells to be formed. And then women also have a menstrual cycle, so there's a certain amount of blood from that standpoint. So there will always be a difference. So when I look at RBCs, and that's one of the ways that's how this will often be written down as RBCs, red blood cells. They'll be lower. Hematocrits, based off of the amount of red blood cells per total amount of volume, will be lower. Things that we have floating around in our red blood cells, called hemoglobin, will be lower. So any lab test that involves red blood cells will be lower for women than for men. I'm talking about healthy individuals. I'm not talking about somebody who has a trauma activation and has lost, 3 liters of blood, and he's a man and go, I thought you said that all men were going to have more. Okay. Healthy people, that's how it works. Men have higher amounts than women. Now, the second part of this will be our white blood cells, leukocytes. And I'm just introducing these. I'm just walking you through kind of where we're going to be going for the lecture tomorrow and on Thursday. What you should notice about this, so my red blood cells, and keep in mind, just again to drive a point home, a lot of the indices when we look at them to make sure that we're comparing apples to apples, we use a similar amount of blood. So millimeters cubed. It's also considered a microliter. Okay, to give you an idea, a microliter probably contains, so 1 drop of blood probably has about 50 microliters in it. So if you think about 1 microliter having 5.5 million red blood cells, think about a drop of blood is this times 50. Okay? So, red blood cells we have in the million. White blood cells, we have in the thousands. Much, much less. This has to do with our immune system. So we're going to spend time going through how they function with the immune system. And then the last component when we talk about structures are platelets. And the one thing I will kind of introduce you to today so you can start putting it in your head is platelets are referred to as thrombocytes. So whenever you hear the term cyte, what should you be thinking? Cell. Platelets are not cells. Platelets are fragments of cells. Okay? So it's a little misleading when we call them thrombocytes. But they are just fragments. Okay? And then once we talk about all of these, we're going to talk about hemostasis. And again, you notice up there it does not say homeostasis. Homeostasis is a steady state situation that every body system basically exists in. It's a window. And I will always try and keep that body system in a particular window. Hemostasis is clotting. And then probably one of the more complicated ones is going to be how we end it, which is going to be our ABO system. And ABO has to do with our blood type. Are you type A, are you type B, are you type AB, are you type O? Are you Rh positive, Rh negative? What does that mean? What are some of the problems that exist with that? Okay, so that's basically what we're going to be talking about the next 2 days. Any questions? Okay, so get into your groups. You need to be in groups at least 3, no more than 4. The first priority